Hey guys, I am back and we are going to be covering DNA replication as well as mitosis today. So we've got a lot to cover. Feel free to speed this video up if you need to, but we wanted to make sure that we got all of this in today. So we've got several standards of excellence. We're going to be looking at SB2 and SB3 today. So SB2 is to obtain, evaluate, and communicate information to analyze how genetic information is expressed in cells. We're going to focus on SB2A and B. A is to construct an explanation of how the structures of DNA and RNA lead to the expression of information within a cell via the processes of replication, transcription, and translation. And we're going to focus on replication today. And then we're also going to be talking about mitosis just a little bit with developing and using models to explain the role of cellular reproduction, including binary fission, mitosis, and meiosis in maintaining genetic continuity. And then we're also going to look at SB3, which is obtain, evaluate, and communicate information to analyze how biological traits are passed on to successive generations. So with SB3, you should be able to construct an argument to support a claim about the relative advantages and disadvantages of sexual and asexual reproduction. So let's start off with a quick review of replication.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented.
All right, guys. So that's basically kind of our background on replication. Whoops, sorry about that. So what happens is that all cells need to copy their DNA or replicate their DNA. And during that process of replication, we've got several enzymes that have different functions. One of them is helicase. Helicase unzips the DNA strand and it unwinds the DNA helix. And the nitrogenous base pairs, they are basically pulled apart or cut from one another. And that happens at the replication fork. Let me turn on my pointer. So right here, the replication fork is just the point where helicase is going through and splitting everything apart. And this exposes the two halves of the DNA strand so that another DNA strand can be um, made, can be created, and that gives us new strands through replication.
So, DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine.
All right, guys. So we mentioned before about A and T pairing with one another and C and G pairing with one another. So we're going to talk a lot more about that in live class. But there are several enzymes used in replication. There's helicase, there's polymerase, there's ligase, there's a few others, but the most important ones that we really want to know about are helicase. That's the one you can see a big blue piece right here. It comes in to unwind the DNA. It's typically called what the one it's referred to as the one that unzips the DNA. And then we have polymerase, and polymerase is kind of like the builder. Polymerase is the one that comes in and starts bringing in the nitrogen pairs. So it brings in the new bases to pair with the existing ones. So this one's going to be the one that, to me, I think of polymerase like a cheerleader. It's standing on the sidelines yelling, "Give me a G, give me a T, give me an A," etc. And it brings them in and it pairs them up on those original strands. The enzymes bond the nucleotides together to form a new double helix. Now, this polymerase is also really important because it goes back and it reads what it did. So it acts like a proofreader to check for mistakes in replication. If polymerase didn't exist, there would be lots of mistakes that happen in replication.
I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in.
All right, guys. So, our cells need a copy of our DNA because of the processes of mitosis and meiosis. Those processes are cell division when we produce new cells. Mitosis happens in organisms that perform asexual reproduction or in organisms that need to grow and develop. So, this is going to be a process where you create an identical copy. Meiosis happens only in organisms that reproduce sexually, like animals. And in this case, we're creating a copy that has half of the amount of chromosomes as the original. So we're going to continue on and talk a little bit more about mitosis now. So, first off, all cells in our body are specialized. They all have a full copy of our DNA, but they all have different jobs. For example, we've got a stem cell down here, and that stem cell has different portions of DNA turned on to become a fat cell, or to become a muscle cell, or to become a sex cell, or to become an immune cell. But then there are other things that are turned off in the DNA. This allows for them to perform different functions. But we have to somehow get this many different cells. And the way that we get hundreds of thousands of cells in our body is through is through Mitosis.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division,
All right, guys. So we're going to talk about and watch the steps of division with the amoeba sisters in just a second. But we need to know that single-celled organisms and some multi-celled organisms do reproduce asexually through a process called mitosis, which is just simply cell division. And in mitosis, what happens is the DNA gets replicated, so we get an extra copy of it, and then each daughter cell receives a copy of the DNA. Mitosis keeps our number of chromosomes constant. That's really, really important. That constant from one cell generation to the next allows us to maintain genetic continuity. And we're going to talk about genetic continuity more in class today. in class today. In a lot of our multicellular organisms, our cell division, mitosis, allows them to grow larger, to develop from a single cell to many different cells, to make other cells that will allow them to replace old and worn out cells or to repair damaged cells. So, mitosis can be very, very important, whether we're talking about a single celled organism that's going to reproduce completely by mitosis, or we're talking about a multi-celled organism that's got to grow and develop and change over time. So you can see down here some pictures. These pictures are going to be important. We've got interphase. That's where, as they said in the video, the cell spends most of its life doing its normal things, growing, developing, copying the DNA. Everything that happens during interphase is all that normal maintenance type stuff. Then we get into the division of the nucleus, which is mitosis. So our first stage is prophase. You can see here in prophase, the nucleus, the chromosomes are beginning to condense. Then we have prometaphase, the nucleus begins to break down, everything starts to do its jobs. Metaphase is stage 2. So prometaphase is kind of an intermediate phase. Metaphase is stage 2. All these chromosomes are lined up in the middle. Then we have anaphase, that's stage 3, where they're being pulled apart from one another. And then telophase is stage 4. That's where the nucleus reforms. And cytokinesis is the final thing. It's not actually part of mitosis, but we talk about it in terms of mitosis because it's actually when the cell itself divides. So let's check out what the Amoeba Sisters have to say about those stages.
Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends, kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells.
All right, guys. So let's check out our mitosis machine here. We start with interphase. And again, you're going to need to know the order of these. You're going to need to be able to recognize the stages in pictures and put them into order potentially. So interphase is first. It will always be the beginning. Then we move into prophase. Prophase is where everything condenses. You start to actually be able to see those chromosomes. The nuclear envelope begins to break down. By metaphase, that nuclear envelope is completely gone and the chromosomes have lined up here in the middle of the cell along that metaphase plate. So again, like they said in the video, meta starts with an M, as does middle. That's how I remember that stage. Then we have anaphase, just like they said in the video. Anaphase starts with an A, and apart or away starts with an A as well. And that's what's happening here. We're pulling the chromosomes apart from each other or away from one another to the opposite sides of the cell. And then telophase and cytokinesis are typically talked about in the same little picture because telophase is when those nuclei begin to reform. You get two new nuclei, and then we get our what's called a cleavage furrow, or in plants, a cell plate, where a basically the cell splits there in the middle. And that's cytokinesis when we actually wind up with two identical cells at the end. So let's talk about DNA and chromosomes real quick, just for a minute. So chromosomes are the DNA that's been all coiled up in the nucleus, and they contain genes and also a little bit of protein. Those chromosomes um, are going to have multiple forms. Those genes can have multiple forms. They're called alleles. And alleles are when you have different copies of the chromosomes. That's the gene that codes for something. We always get a copy of the chromosomes in organisms that are sexually reproduced, one from mom and one from dad. One always comes from the female parent and one always comes from the male parent. And our genes, our DNA, contains that coded information that's needed to make proteins. So remember, DNA is super, super, super important. We're going to use that DNA to make proteins. Proteins are so important to our bodies. And those genes have to be divided equally into the two new cells. They have to be replicated at the beginning, and then they have to be split up so that there are two equal cells that are identical to one another at the end. All right, guys, that is everything I have for you for this video. I hope that you guys are having an amazing day, and I will see you guys soon for our next flipped recording. Bye.